This video is a part of the gram-positive bacteria identification. This one going to deal with mannitol salt agar, MSA. MSA is a selective and differential medium for the isolation and differentiation of pathogenic Staphylococcus aureus. MSA is selective as it contains 7.5% of salt. Seawater contains about 3.5% of salt. This makes MSA very hypertonic. As a result, most organisms cannot grow on MSA. Staphylococcus aureus is known to be a halophile and can tolerate and grow on higher concentrations of salt. MSA is also differential. MSA contains the carbohydrate mannitol. Mannitol is a sugar alcohol with the chemical formula of C6H14O6. It acts as a sweetener, although it's not well absorbed by the intestine. Also, few bacteria have the ability to use mannitol. Some can ferment it. Staphylococcus aureus can ferment mannitol. The reaction is a typical carbohydrate fermentation with the production of excess hydrogen ions. These cause the pH of the medium to drop, become acidic. MSA also contains the pH indicator phenol red. This is the same indicator that is used in the carbohydrate tests of the enteroplury tube. Phenol red will impart a bright pink color at a pH above 8.2. It will also have a yellow color at a pH of less than 6.8. After inoculation, a positive result is recorded with a change in color from the pinkish color to a yellow color. Inoculation is done by taking a sample, which could be either a biological sample from a wound or an abscess, or a sample from a pure culture. So here's the procedure. We will apply 2 different bacteria for comparison. We mark a line to divide the plates into 2 halves, and we label the plate. We're going to use sterilized cotton swabs for this demonstration, but this can also be done using sterilized loops. With the sterile swab, we pick up a small amount of one bacteria and inoculate it on the medium, making a zigzag. Then we dispose of the cotton swab. With a new sterilized cotton swab, we pick up a small amount of the other bacteria and inoculate it on the medium, making a zigzag. Then we dispose of the cotton swab. Next, we seal the plates with parafilm. If we are using a sterilized loop, we would use the same standard procedure we've done before. First, flame the loop, let it cool, pick up a small amount of one sample, then inoculate it on the MSA. Flame the loop again and let it cool down. Pick up a small amount of the other bacteria, then inoculate it on the MSA. Flame the loop again and let it cool down. The loop can now be stored and the plate sealed with parafilm. Invert the plate and incubate it upside down for 12 to 24 hours to see the results start to show. We're going to incubate this on a light box so we can film the growth.